So in this video, I want to discuss the different reactions that we see in laboratory 30 for these carbohydrates. The first one that we're going to encounter is called the Benedict's test, and it says it's for testing for reducing sugars. And what this means is that there's a sugar molecule that has an aldehyde group up top there that can undergo this oxidation reaction. Now, we won't actually see this change from the sugar to the next. What we actually visualize is this color change from blue over to a red color. These reactions occur very quickly. When we actually watch the videos, what you'll see is that it's going to last for a very short period of time. It doesn't take much time at all for these reactions to take place. And what we'll do is we'll watch the Benedict's reaction in just a minute. First, let's look at the report sheet. In part A, we see there's the Benedict's test. And in the video, you'll see each one of these labeled pretty well, I think, including an unknown. And so you'll want to record the information for the unknown. In the observation section, really, you're just putting red or blue. That's as simple as I can put it. You're just saying, what is the color after the reaction takes place? One way to keep track of this, reducing sugar, red, turns a red color. Okay, that's that copper oxide solid that forms. Now, before we watch the video, let's just go ahead and do a quick review of reducing sugars, just for a moment. So when I talk about a reducing sugar, what I'm really discussing is something that can undergo mutarotation. That's the way I view it. Now, just a minute ago, we saw the Fischer projection for glucose. Well, as we learned in the lecture videos, which you should really watch before doing this lab, one of the anomers for glucose is this guy. And this is beta-D-glucose. Right, the majority of the molecules of glucose in a solution form these cyclic structures. Very few are actually in that, what I call the open chain structure of the Fischer projection. But it can still undergo the Benedict's. It could still give a positive test for a Benedict's reaction, even though there's no aldehyde group on the structure, because what you have to realize, what you have to remember is that this Hayward structure can open up and then form the D-glucose molecule as we see in the Fischer projection. This is actually the molecule that turns red, that reacts with the Benedict's reagent. That's why we call it a reducing sugar. But again, notice what it's doing. It's really, in my mind, a tool, a reaction to say yes or no, mutarotation is occurring or not. You can also have reducing sugars that are disaccharides, such as this alpha maltose molecule. And notice I'm not going to take any time to break down the details of this structure. Oops, that's wrong. There we go. Again, it's a really good idea to watch the videos before you do this lab. So right here's a structure for our alpha maltose. And what we have to realize is that this number 1 position is this carbon here. That's the aldehyde group. So when this number 1 position is not bonded to anything, meaning it's just a simple sugar and hydroxy group attached to it, then it undergoes this mutarotation reaction. In alpha maltose, what you see here is that number 1 position is locked because it has the glycosidic bond here. Whereas that number 1 position is, I'll just say, unlocked. And so again, it should turn red in a Benedict's reaction. Indicates that we have one of these number 1 carbons, really. We have this carbon and ring structure that can open and close to allow the aldehyde group to show up to then react with the Benedict's reagent. So as you're watching the video, sort of keep that in mind as you watch the ones that change the color from red to blue. I'm sorry, blue to red.